Hey guys, so today we're going to be looking at animal development and discussing the embryonic stages. Specifically, we're going to focus on the first stage in the embryonic stages, which is fertilization. As you can see here, there are many common features in early embryonic stages among animals, and this is seen through model organisms, which have been used to study these developments. Just to let you guys know, model organisms are organisms used to understand certain biological processes and other concepts. So again, today we're just going to be looking at and focusing in on the first stage in the embryonic stages, which is fertilization. Okay guys, so the first part of the embryonic stages is fertilization. What is fertilization? Fertilization is the fusion of haploid gametes, specifically sperm and egg. Now it is important to note that the fusion of these haploid gametes, they produce a diploid zygote. Now, sperm will break down the protective layer around the egg. This protective layer is known as the zona pellucida. So as you can see here, you have your sperm, you have your egg, and you have your jelly coat, which is the zona pellucida. So what will happen is once the sperm breaks through the zona pellucida, receptors on both the sperm and egg lock, and a hard coating forms around the egg, preventing polyspermy. So as you can see in this bottom left-hand corner, these would be the two receptors that will meet together. So these two will lock, and polyspermy will be prevented. And in the next part, we're going to talk about how this happens. Also, we're going to talk about how the sperm breaks down the jelly coat, or the zona pellucida. So here on the left-hand side, we have the sperm again, and the egg and the jelly coating surrounding it, the zona pellucida. Now, at the end of this sperm, we have the acrosomes, and the acrosomes are hydrolytic enzymes that will break down the zona pellucida. So here you'll have the acrosomes will be released, and they will surround or go towards the zona pellucida. And because there are millions of sperm doing this and breaking down the zona pellucida, yes, the sperm will die off, but because there are so many of them, there will eventually be one that reaches the egg and fertilizes it. And again, these hydrolytic enzymes break down the zona pellucida. And once the sperm reaches the egg, you then have the acrosomal process, which is shown here. And the acrosomal process is the elongation process where the sperm and egg receptors meet. So here's the sperm receptor, here's the egg receptor. And they act as a lock and key model which fuses the membranes together. So again, this fusion right here is known as the lock and key model. And once the receptors come together and they fit, and because they fit perfectly, the fusion of the egg and sperm plasma membranes occur, causing the fast and slow block to polyspermy. And polyspermy would be the fusion of multiple sperm with the egg, but due to this process right here, and the fast and slow block, it actually stops polyspermy. Okay, so once the fusion of the eggs happens, first off is the fast block. Once the fusion of the sperm and egg membranes happen, you have calcium rushing into the cell. And this rush of calcium actually causes a neutralization in the egg cytoplasm. And this neutralization in the egg cytoplasm actually stops the attraction of other sperm and helps with the prevention of polyspermy. So as you can see here, here's the egg, right? Again, once the fusion happens, so you'll have, a fusion of the sperm fusing with the egg's membrane, and you'll have calcium rushing into these channels, right? So you'll have them all rushing in, and because the inside of the cell is negative, right? So these are all negative signs. The positive ions of the calcium rushing in neutralizes the egg cytoplasm. Again, the cytoplasm, the inside contents of the cell, and it causes the neutralization of the cytoplasm, causing other sperm to be less attracted to the egg. So shortly after the fast block, we have something known as a slow block, and this occurs a few seconds after the sperm binds the egg, and certain vesicles known as cortical granules, which are seen here, will actually release their contents within the egg cytoplasm. So here, these cortical granules sit beneath the egg membrane, and when the fusion occurs, they're going to release their contents all throughout the cytoplasm, exocytosis into the cytoplasm. And this exocytosis of these contents actually causes the formation of the fertilization envelope, which is a hardened, almost shield that surrounds the egg and prevents polyspermy further. And this process is known as the cortical reaction. Now, it is important to note that the slow block will occur in all different species. However, the fast block will not occur in mammals. It has never been found to be occurring in mammals. The fast block occurs in species such as sea urchins, but again, it has not been found that this occurs in mammals. Only the slow block will. And lastly, for the cortical reaction, it is important to note that the cortical reaction needs a lot of calcium in order to take place. It is also important to note that it is possible, the cortical reaction is possible because of the neutralization of the egg after the calcium rushes in. Again, without the cortical reaction, there is no long-term prevention to polyspermy. So before I go, I just want to say thanks for watching. Please like, subscribe, and leave any questions in the comments in regards to this video or any videos you guys would like to see in the future. Thank you.